Hello everyone, I am Kanchana working in SNS College of Engineering as an assistant professor in the Department of Chemistry. In this video now I am going to say about instrumental methods of analysis, potentiometry. The principle involved in the potentiometry is when the pair of electrode is placed in the sample solution. It shows the potential difference by the addition of the titrant or by the change in the concentration of the ions. Potentiometry is when the pair of electrodes is placed in the sample solution. It shows the potential difference by the addition of the titrant or by the change in the concentration of the ions. The reference electrode is the electrode which contains of its own potential value and it is stable when dipped into sample solution. Here analyte solution is the solution whose potential is to be measured. The indicator electrode is the electrode which responds to change in the potential of analyte solution. The electromotive force of the complete cell is given by the following equation. E cell equal to E reference plus E indicator plus E junction, where E reference is the electromotive force of reference electrode. E indicator is electromotive force of indicator electrode. E junction is electromotive force at the junction of the liquid. E cell equal to E reference plus E indicator plus E junction. The main theory involved in the potentiometry is when the known potential electrode immersed in the sample solution, then the potential is given by Nernst's equation. E equal to E naught plus 0.0592 divided by N into log C, where E is the potential of the solution, E naught is the standard electrode potential, N is the valency of the ion, C is the concentration of the sample solution, 0.0592 is the value obtained from the RT divided by EF, where R is the gas constant, T is the temperature in Kelvin, F is the Faraday's constant. Electrodes. These are mainly used to measure the voltages. Mainly two electrodes are used in potentiometry. They are follows. Reference electrode, indicator electrode. Reference electrode. These are mainly used for the determination of analyte by maintaining the fixed potential. For example, standard hydrogen electrode, silver chloride electrode, saturated caramel electrode. The reference electrodes are classified into two main classes. They are as follows: primary standard electrode, standard hydrogen electrode. Secondary standard electrode, for example, silver chloride electrode, saturated caramel electrode. Potentiometry. Indicator electrode. It is used to measure the potential of analyte solution comparing with that of reference electrode. Its potential is directly proportional to ion concentration. For example, hydrogen electrode and glass electrode. Antimony oxide electrode. There are two classes of indicator electrodes. First one, metal indicator electrodes, and another one is ion selective electrodes. Metal indicator electrodes. These develop electrical potential in response to a redox reaction on the metal surface. Platinum or gold are used as metal indicator electrodes. And ion selective indicators. These are composed of ion selective membrane by which the ion crosses and it creates the imbalance. For example, glass membrane electrode. Types of potentiometric titrations: acid base titration, redox titration, complexometric titration, precipitation titration. Clinical chemistry: ion selective electrodes are present sensor for clinical samples because of their selectivity for analyte in complex matrices. The most common analytes are electrolytes such as sodium, potassium, calcium, and hydrogen and chlorine and dissolved gases such as CO2. Environmental chemistry for analysis of CN, cyanide, ammonia, and NO3 and F3 in water and wastewater. And potentiometric titrations for determining the equivalence point of an acid base titration. Potentiometric titration for determining the equivalence point of an acid base titration. Possible for redox, precipitation, acid base, complexation, as well as for all titration in aqueous, non aqueous solvents. Agriculture: NO3, NH4, and calcium, potassium cyanide, chlorine in soils, plant materials, feedstuff, and fertilizers. Detergent manufacturing: calcium, barium, fluorine for studying effects in water quality. Food processing: salt content of the meat, fish, dairy products, fruit juices, and solution. Calcium in dairy products and beer. Potassium in fruit juice and wine making. Corrosive effects of NO3 in canned foods. Fluorine in drinking water and other drinks. NO3 and NO2 in meat preservatives. Thank you.